fácil transmitir para o YouTube. Aí.
Beleza.
So let's switch to English now. Welcome everyone who joined us for the seminar today. Good afternoon and good morning for Raíssa in Australia. Today we are pleased to have here with us a former student of our postgraduate program, Dr. Raíssa Fonseca. So a quick introduction before she starts. Raíssa did her PhD here in Brazil at the University of São Paulo at the lab of Professor José Maria Mozig, also known as Professor Pepe. And also she went abroad for a period at the University of Minnesota. And in her thesis, she researched the pathogenesis and regulation of the immune response against Chagas disease, where she found T cells infiltrating the heart of infected mice. At this time, she also started to work on tissue-resident memory T cells, or TRMs. And nowadays, she's still doing beautiful work with memory T cells responses as a postdoctoral fellow in Professor Laura McKay's group at the Doherty Institute at the University of Melbourne, Australia. So thank you so much, Raíssa, for being here with us today. For Raíssa, now it's 7 a.m. in the morning, and she dedicated her time to be here with us. So thank you so much again, and now the floor is yours.
Thanks for the introduction, Luisa, and thanks for the invitation to present. I gave my talk, a pretty generous talk on this topic, but I am going to present data from our last published paper. So this is the institute I work at. It's Peter Doherty, which is part of the University of Melbourne. And I am at Laura's lab working on this project on the last 4 years since I came here in 2018, and it finally got published earlier this year. So I am going to try to focus a little bit on tissue-resident memory T cells, but I did a very simple introduction here to try to catch people who are not so familiar with the field. So if you imagine here a naive host where known previous mounted adaptive response has been triggered against this pathogen, you're going to have no or a very small amount of specific T cells to that pathogen. So this infection is going to start to progress, and in this case, in a viral infection, you're going to see this rise in viral particles once the virus starts proliferating inside of cells, while the number of T cells here is very negligible. And importantly, these naive T cells are going to be recirculating in lymph nodes, for example, and they're basically screening for their cognate peptide. So they're screening for their antigen, trying to find cells bearing the peptide so they can expand and get activated and then respond to this infection. So in the lymph nodes and spleen, you're going to find both naive CD4 and CD8 T cells. And once this dendritic cell drains from the tissue where the infection started and migrate to the lymph nodes, these naive T cells are going to get activated and are going to respond with a high proliferation and cytokine production, giving rise to this heterogeneous effector T cell pool that will then migrate in a process called expansion, that will then migrate to these barrier tissues and try to control the infection. Once these cells reach the barrier tissue, they're going to respond to control the infection, and they're going to enter a phase called contraction once the pathogen is eliminated. Once the cells contract, they're going to leave behind a population of circulating memory T cells and tissue-resident memory T cells that will reside in this barrier tissue and are going to be the primary source of contact with this pathogen again once a reinfection occurs. So this population of memory T cells are long-lasting, and they're known to be prevalent for years. In the mouse case, you are going to find memory T cells that are actually long-lived to the lifespan of the mouse. So if we move on from here and focus on the tissue-resident memory T cells, I would like to point some of their main characteristics. So these cells are non-recirculating. They are going to live in these barrier tissues, and they were shown to provide better outcome in the case of cancer and infections. So these cells will be in these peripheral locations, being the first responders in that in any of those situations. However, due to its characteristic of residing in barrier tissues, when they recognize self-antigens, they can cause autoimmunity, and they were shown to be present in those situations as well. And because these cells are part of this major network that will be related to infections, cancer, and autoimmunity, they have been a focus of new immunotherapies, such as adoptive cell transfer by modification of the T cell receptor. So these cells can respond better to cancer, for example. And part of the new vaccine strategies that are trying to increase the generation of the tissue-resident memory T cells in the barrier tissue, promoting a better first contact with its pathogen. And that's where most of our research is directed to. However, I would like to remind you that we are doing very basic T cell development research to try to inform those immunotherapies and vaccine strategies. So these cells are found in all organs, and especially I'm going to highlight tissues that are contact with the external environment, for example, the lungs, and the intestines, and the skin. And here they're going to coexist with C4 T cells. However, we are going to see that there is a discrepancy between the cells that are in this epithelial barrier and cells that are in the sublying tissue. So CD8 T cells are more dominant in this barrier tissue. They are also going to be existent in organs that are high vascularized, such as the liver, for example. And we are also going to find both CD4 and CD8 T cells there. However, both present population that is resident, and they're going to coexist in organs with high circulation with the circulating memory T cells. And they are also interestingly found in secondary lymphoid organs, such as lymph nodes, where we can find a population of resident T cells that are not super described yet, but some of them at least are believed to be coming from the periphery. And they're distinct from the circulatory memory T cells by its phenotype. So they're going to express a different set of molecules, different transcriptional signatures. So the genes expressed are going to be different from that T-cell population. Interestingly, they do present a different morphology. So because these cells are found in this epithelial layer, they are going to have to kind of squish themselves through the epithelial cells, making them look dendritic-like, which I'm going to show you later on. And the development of these tissue-resident memory T cells and T-circ are also different. And most of our new research has been done in this area, trying to find which cells can actually generate circulating and resident memory T cells. So to study these tissue-resident memory T cells, two main techniques were utilized so far, and this informed a lot of the phenotype and the transcriptional signature that we can now use to investigate in our models. So transplantation is one that we can use in both mouse models and humans. So here what happens is that if we use an immune mouse, which has tissue-resident memory T cells in the skin, for example, we can remove that tissue and transfer to a naive mouse, which didn't have any tissue-resident memory T cells or circulating memory T cells from the specific T cell pool. So once this tissue is transplanted, you're going to see that cells that are non-resident, they're going to fall back in the circulation of this naive mouse, while the resident memory T cells are going to be residing in that patch. So they're not going to leave the skin, the transplanted skin. So then you can define the cells that are host-derived or cells that are donor-derived from that tissue. If you want to take a more systemic approach, then you can use a model called parabiosis, where we join the circulation of two mice here. So the way you do this is just to make a small cut in the skin and tie both mice together, and the circulation will heal as a joined one like that. And then what you're going to have is that in every organ where tissue-resident memory T cells were generated, they're going to be exclusive in the immune mouse, while cells that are circulating, they're going to be present also in this partner, in this naive partner. So then you can define and find which populations were resident or not. And a lot of this work has been done by David Mazepou's group and published by Lily in 2019 here, and by Elizabeth Steiner before, showing that most of the cells in the small intestine and large intestine, and here the female reproductive tract, are mostly resident memory T cells. So very few cells are actually circulating at the memory stage in that organ. While in organs that are highly vascularized, then you're going to see a lower proportion of tissue resident memory T cells, and they're going to be coexisting with circulatory memory T cells. One thing that I would like to point and to highlight from this resident population is that if we split these cells between CD4 and CD8 memory T cells, here we have an example where similar proportions of CD4 and CD8 memory T cells are in equilibrium. So only 5% of CD4 and CD8 memory T cells from the female reproductive tract are actually in circulation. So they're equilibrating with the circulation. But when we look at other organs, as I mentioned before, these CD4 memory T cells are now equilibrating with the circulation, while CD8 memory T cells are not. So it looks like these CD8 T cells are more resident, if you like, than the CD4 population. But when we move to these highly vascularized organs, then we see that this tendency changes a little bit, with more CD8 T cells becoming equilibrating, while the CD4s are doing it as well. So it looks like there's a divergence between epithelial organs and highly vascularized organs. We can look at the phenotype of these cells and see the major differences between the CD4 and CD8 memory T cell population. And what I would really like to highlight here is this double positive population for CD69 and CD103 inside of the CD8 memory T cell pool. And this was shown in humans, connecting to our, generated knowledge in mice. We are now translating that and finding this population of tissue resident memory T cells in humans. And what these two molecules are doing, CD69 and CD103, is promoting anchoring to this epithelial cell, for example, and other tissues in the body, anchoring this T cell to the periphery. And in the meantime, CD69 is antagonizing the S1PR receptor, making these cells to reduce trafficking and promoting tissue retention. And now I'm going to show some of the results we got in this latest published paper. And I would like to thank Tom Byrne for all the help finishing this project. And his dog's name is Farofa, because she has all the patches like Farofa. So just to follow up here, we're going to use the skin as an example of an epithelial organ, right? So we are using a mouse model of skin infection, and we do that using HSV. So if you compare here a mouse that had tissue resident memory T cells or not, you can see that the lesion caused by the virus in the skin is a lot less pronounced in mice that had tissue resident memory T cells versus mice that didn't have that protection, that local protection. So you can see that the viral infection here is a lot worse in the skin. When we zoom in and look at the organ using microscopy, we can see that both CD4s here in red and CD8 here in pink are going to be both in the skin dermis and the skin epidermis. So these T cells actually distribute globally in the skin. But at the memory stage, what we see is that the CD8 memory T cells are going to be in the epithelial layer, while CD4 memory T cells are going to be mostly in the dermis. And previous research have shown that the CD4 T cells are more in equilibrium with the blood than the CD8 T cells, which are actually resident. So we wanted to investigate more about those differences between CD4 and CD8 T cells and try to see how can we make, or how does this resident population actually is generated. So here we can look at some transcription factors, and I'm going to highlight ranks 1 and ranks 3 and show this expression of CD69 and CD103, which we have previously seen in the last slide. So to start with these molecules, we can see that the CD8 tissue resident memory T cell population does express CD103, while both express CD69. However, CD103 is absent in this CD4 resident memory T cell population. If we look at these transcription factors, first ranks 3, we can see a high expression of ranks 3 in CD8 tissue resident memory T cells. And this is very interesting because ranks 3 was previously shown to be participant in the generation of resident T cells. But this expression of ranks 3 is antagonized in CD4 T cells, at least at the naive stage, by THPOC, which is going to help with the timing selection, right? So ranks 3 is the transcription factor that's needed for naive CD8 T cells, while THPOC is needed for CD4 T cells. But we find this other transcription factor from the ranks family here that is ranks 1, and that's actually upregulated in the CD4 T cell pool. However, it seems to be decreased at the tissue resident cell stage, but it's coexistent with ranks 3 in CD4 T cells, while the expression of ranks 1 is now low. But I think that the next slides are going to make sense of why and how is ranks 1 and ranks 3 participating in this generation of residency. So to investigate more of the role of those transcription factors, what we did was to use CRISPR to knock out ranks 1 and 3 and look at memory stage. How are these cells looking in the skin? So here we can see that there is no change, no major change in the population generated after editing ranks 1. So in the absence of ranks 1, it seems like similar numbers are still becoming resident in the skin. However, when you delete in CD8 T cells ranks 3, we see this major decrease in the generation of residents in the skin. So ranks 1 doesn't affect CD8s, but ranks 3 does. And very interestingly, when we look at CD103 expression, we see this major decrease in the expression of 103, which is partially why this population is impaired. So basically, CD8 T cells require ranks 3 to establish residency. And that was previously shown by Justin Mueller and Ananda Goldrath. But when we now go and look at CD4 T cells for the need of ranks 1 and ranks 3 to generate memory T cell, we see the inverse. So we see that ranks 1 is affecting CD4 T cells, but ranks 3 is not. And we now don't see any difference in the expression of CD103 when we knock out those two transcription factors. So how does this interplay with the generation of tissue resident memory T cells? How is ranks 3 assisting the generation of these memory T cells in the epithelial layer? So to investigate that more, we use the inverse to, instead of knocking out, we are promoting overexpression of this transcription factor, both in CD8 and CD4 T cells. So first, if we isolate and look just at CD8 T cells, we can look at CD8 T cells that are overexpressing ranks 3, and that will increase the generation of tissue resident memory T cells in the skin, but that will not affect as much the circulating memory cells. And we can increase the expression of CD103, which we saw it does help tissue retention. So by forcing ranks 3 expression, you can increase CD103 generation and the CD8 T cell residency. What does happen when we do that in CD4 T cells? So that's Mueller's data showing that also happens in the intestine. And he also showed that in tumor studies, true, that will increase the generation of tissue resident memory T cells there and increase protection. So if we go ahead and look at our CD4 T cell population when we overexpress ranks 3, what we see is similar tendency. So we see that there is an increase in the generation of the population of resident cells in the skin. And we see, an increase in the proportion of these cells in the epidermis now, while before control cells were more prevalent in the dermis. So what RANKS3 is actually doing in the CD4 T cell population is to increase the residency specifically in the epithelial layer. But how is RANKS3 actually modulating residency? How does this occur? So to investigate this further, we looked at the transcriptome of these populations. So here we did a co-transfer of these three main populations. So CD4 control cells, CD4 T cells overexpressing RANKS3, and CD8 control cells. And we transferred those cells to HSV-infected mice and recovered the cells from the skin at memory. What we found here was that even though this CD4 T cell population overexpressing RANKS3 had increased expression of RANKS3, they didn't become CD8 T cells. So they are still distinct, but they also are distinct from this control CD4 T cell population. If we look at the genes that these populations are expressing, we can see common genes between CD8 T cells and CD4 T cells overexpressing RANKS. So they're, even though they're transcriptionally distinct, they do share some common genes. If we go ahead and look at tissue-resident memory T cells related genes based on previously published data, we can see that this CD4 T cells overexpressing RANKS3 and CD8 T cells, they are actually regulating the residency genes in a very similar fashion. And this is in comparison with the expression in CD4 control cells. So it looks like there is similar regulation of this residency-associated genes in CD8 and CD4 RANKS3 population. So we wanted to investigate how does that relate to a transcriptional signature of tissue-resident memory T cells. And to do that, we used the previously published data set from Laura's group, where we can isolate circulatory memory T cells and tissue-resident memory T cells. And then, like we did for CD4s and CD8s, we can look at the common genes and the exclusive genes for TCERC and for TRMs. So once we select these genes that are exclusive of tissue-resident memory T cells, we can select the genes that are the top most down-regulated genes in skin tissue-resident memory T cells, or the most up-regulated in that population. And then we can look at those down-regulated and up-regulated genes from skin TRM in our CD4 T cell populations. If we plot all the genes that are upregulated or downregulated in skin TRM cells, we can see in our chosen population, so CD4 RANKS3 and CD4 control, genes that are upregulated in the CD4 T cells overexpressing RANKS3. And we can see that there is many common genes that are upregulated in skin tissue-resident memory T cells and in our CD4 RANKS3 population. And the same is true for the genes that are downregulated, right? So that creates a positive correlation of the genes associated with residency and the genes being transcribed in our CD4 RANKS3 population. How is RANKS3 inducing this TRM signature, though? How is this happening? How is this being controlled by RANKS3? So to answer that question, what we did was to look at chromatin accessibility in cells overexpressing RANKS3. So we did ataxic in cells, in CD4 control cells, in CD4 T cells overexpressing RANKS3, and in CD8 control cells. And here we can look at the footprint of RANKS3 in these populations. And we can see here in the green line and the purple line behind it that the occupancy of RANKS3 is very similar between the CD4 RANKS3 and the CD8 population, while less occupancy is seen in the CD4 control population. If we go ahead and look at gene loci that are TGF-beta regulated, we see an increase in those two populations, so CD4 rank 3 and CD8 T cells, in relation to the CD4 control population. So it looks like rank 3 is promoting more accessibility in these TGF-beta regulated regions. And we can pick our favorite, handpick our favorite gene, here CD103, and we can see that the CD4 T cells overexpressing rank 3 have major accessibility in gene regions within the CD103 loci. So it looks like there's similar accessibility between TGF-beta regulated genes in CD4 rank 3 cells and CD8 T cells. And then we can go ahead and create this model of how do we think this transcription factor is inducing this resident-like response. So what we think is going on at this point is that rank 3 is changing gene accessibility, mainly in TGF-beta regulated regions. And that will allow now CD4 T cells to respond to TGF-beta in a similar fashion that the CD8 control cells do, but different to how these control CD4 T cells respond to TGF-beta. And that will induce, will facilitate the induction of these genes, these molecules that are associated with TGF-beta in residency. So to test this hypothesis, what we did was to first do an in silico analysis of our data. And we used the third data set here to look at cells that are sufficient or deficient for the TGF-beta receptor. So these cells don't have the TGF-beta receptor, so they would be lacking the response to TGF-beta. If we color here in yellow all the genes that are regulated by TGF-beta, so genes that are upregulated and downregulated by TGF-beta, we can look at that in conjunction with these genes that are present in tissue-resident memory T cells in the skin and in our CD4 rank 3 population in that quadrant. So isolating here the bottom quadrant, what we see are genes that are TGF-beta regulated, right? So those are the painted ones in wild-type cells, downregulated. We have genes that are downregulated in skin TRM cells and downregulated in our CD4 rank 3 cells. We'll go ahead and look at the top right quadrant. Now we are seeing genes that are upregulated, creating here a correlation, a positive correlation, indicating that these three aspects are positive correlated. So what this indicates is that rank 3 is actually promoting large-scale transcriptomic changes that are highly correlated with those changes seen in skin TRM cells, and mostly driven by TGF-beta, it seems like, and we can test that for other cytokines if you like. So how do all these epigenetic changes and transcriptional changes actually related to TRM functionality? Is that actually helping the immune response? What's going on in the skin where we generated that memory population based on rank 3 overexpression? So if we first test this in vitro, we can see that, for example, for CD103, we see better induction of molecules driven by TGF-beta. So we can see that our CD4 rank 3 cells are now able to rapidly upregulate CD103 in vitro. And if we move in vivo, we can validate this need of the TGF-beta receptor to generate this population of tissue-resident memory cell. So I'm just going to talk you through this model because it's a little bit complex based on the number of cells that we are using here. So all the cells in this experiment are CD4 T cells, right? And we are using cells that are sufficient for the TGF-beta receptor, so wild-type cells, and cells that are knockout for this TGF-beta receptor. And we transduced both cells with a control vector. So these are normal CD4 T cells that are either lacking TGF-beta receptor or not. And in a separate set, what we did was to promote overexpression of RANKS3 in wild-type cells in these TGF-beta knockout cells. So these were transferred into different recipients for the sake of using the congenic markers we have available. But what we can see here is that while our control cells are not really impacted by the absence of TGF-beta receptor, in the absence of this receptor, we see no generation or no increase in the generation of tissue-resident memory T cell here for the cells overexpressing RANKS3, meaning that actually RANKS3 does work and induce residency, but that happens in a TGF-beta manner. It does require this receptor. And to validate this model, we also see that there is an increase in CD103 expression, as we did see in vitro in this in vivo model, and that's also TGF-beta dependent in vivo. So now if we go ahead and look at our cells in the skin, here, as I shown you in one of the first slides in the presentation, we see this divergence between CD4 and CD8 T cells in the skin, right? So first details is that CD8 T cells in the epidermis, they have this dendritic-like shape, while the CD4 T cells reside in the dermis or present in the dermis, they're going to have this more round-like shape. And here you can see the distribution of the cells in the epidermis and dermis, as I showed you before. If we now go and look at our CD4 control cells and CD4 T cells overexpressing RANKS3, we see a similar distribution pattern. And we also see this difference in the shape of the CD4 T cells overexpressing RANKS3, more dendritic-like. So they are now resident in the epidermis and they have a dendritic-like morphology. If we go ahead and look at their movement, here on the left side, you can see the CD4 T cells very fastly moving around the dermis, and you're going to see that they are still round-shaped. While the CD4 T cells are expressing RANKS3, they are moving very slowly, if you can find them here. And you're going to see that they are more dendritic-like, as I showed in the previous image. And we can quantify all the information from our video. And what we see is a decrease in sphericity, we see a decrease in velocity, and we see that they're moving less in both the Y and the X-axis in the video, right? And that leads to a decrease in displacement length. So these cells are actually moving less, leading to a change in shape and motility. So what does that lead to? That does lead to a decrease in egress. So that change in movement, if we take a piece of that skin, put that in vitro, and look for cells that are egressing the skin, we are going to see that these C4 RANKS3 cells are now leaving this explant less than the control cells, meaning that somewhat they're more resident than that population. We can also look at their cytokine pattern production. So we can see that these cells are expressing more interferon gamma and TNF, meaning that they're more CD8-like protectants in the barrier tissue. If we go ahead now and use this model to look at the viral challenge, so here we have a group of mice that were transferred in the dermis. So these cells are now in the skin. Those are CD4 control T cells, and a separate group containing these CD4 T cells containing RANKS3. So these cells are only present in the skin. 14 days later, at memory, we are going to challenge these mice with their cognate antigen, right? So we are going to challenge them with a new with a HSV infection and see how are these cells protecting. So first, what we saw was that there is an increase of inflammatory monocytes, NK cells, and very slightly at CD4 and CD8 T cells in the skin, bearing the CD4 control, the CD4 RANKS3 cells, but less so in the control group. So besides increasing the recruitment of these immune populations and increasing cytokine production, we see a big decrease in the viral titer in that group. So it seems like CD4 rank 3 cells are enabling better protection against this viral infection in the skin when compared to CD4 control cells. So here we basically describe what mechanism is rank 3 inducing residency, and how does that actually help in protective immune scenario. So I'm going to talk you through the conclusions of this last set of results. And if we, think of the very basic response, it seems like tissue resident memory T cells, they actually participate in the immune response in diverse scenarios. So present in cancer, infections, and autoimmune diseases, and they can be manipulated to improve disease outcomes. So we have just seen as an example of, let's say, adoptive cell transfer, where we are genetically modifying these T cells in vitro and placing them in vivo in a scenario where they can actually respond to these infections. Another focus of the lab is how to deplete these cells in vivo. So in the case of an autoimmune disease, we are searching how to, or what can we target in these cells to remove that population from the organ where the cell would be responsible for the autoimmune disease. So next, we saw the ranks 1 and ranks 3 differentially programmed CD4 and CD8 tissue resident memory T cell development. And it seems like ranks 3 actually control epithelial residency, and that's done in a TGF-beta dependent mechanism through epigenetic modifications, so increasing chromatin accessibility in those genes. So ranks 3 induces this CD8-like TRM transcriptional signature in CD4 T cells by controlling this chromatin accessibility and the transcription of TGF-beta regulated genes. And it seems like ranks 3 doesn't force a protective CD8 TRM-like immunosurveillance in CD4 T cells in the skin in the model that we utilize. And I would like to acknowledge everyone in the lab who helped with this work, especially Laura and Frank, who were, major parts of it since I started, and Tom for helping me at the final stages of this publication, and Luke, who did most of the bioinformatic analysis for this paper, and everyone else. And I would like to also thank Sapna and Sarah from the Mueller lab with the microscopy data, and Frank and Kayla from the SAT-PAFI lab, who we were major parts of the ATAC-seq analysis in the final stages. And if you're interested in TRMs or any other topics, if you haven't seen this yet, you should definitely check it out. These global immunotalks have really good speakers, and I will highlight these two. So this first one is by Dr. David Mazopoulos, who was my primary supervisor when I was working in Minnesota, and he's an absolute genius of the field. And Laura McKay, who is incredible, and she's a superstar, and she's really good, and her talks on tissue resident memory T cells are simply stunning. And, obviously I wouldn't, there are many other talks in the topic by Donna Farber explaining how these cells working in humans, and Marion Pepper and C4 T cells, and Steve Jameson, and then the Gold Ref and others. So just make sure to check it out if you're interested in this topic. And thanks for listening to the presentation.
Thank you so much, Raíssa. Amazing talk. Very nice data. So we are now open to questions. If you want, raise your hands or open your mic to make a question to Raíssa. And if you let me, I want to ask a question. Yeah. So it's more a philosophical question, because I was thinking about in the beginning when you were talking about the intestine as well. There is also have a big percentage of CD4 cells and CD8 as well. But I was thinking about other resident cells that do not generate memory, the ILCs that are also very important in these sites. So I don't know about the mechanisms that make the cells stay in the sites, and if they are similar to the T cells. Do you know something about the ILCs and how they are?
Yeah. There are, well, we do know that there are plenty of ILC that are resident in tissues, right? And there is a correlation. So if you look at the transcriptional signature of those cells and, you know, the CD8 tissue resident memory T cells, for example, the genes that are controlled, at least the non-related to memory T cells, they're similar. So you're going to still see a decrease of circulatory, you know, molecules and an increase in these anchoring molecules. And we published another study in, I think it was 2020, that shows that this fatty acid binding proteins, which are required for tissue resident memory T cells to survive, are the same between the ILCs and the CD8 T cells residing in organs, indicating that their metabolism might also be similar. And that is organ exclusive. So I didn't focus on that during my talk, but tissue resident memory T cells are extremely heterogeneous. So there are different types within each tissue, but if you look across tissues, they are completely different. So a cell that resides in the liver is completely different from a cell that resides in the skin or in the gut.
And I imagine for other cells that generate memory as well, as we think, if we think about B cells in the intestine, those producing IgA, for example, the mechanism may be very different.
Yeah, exactly. So it seems like there is some shared signature within each tissue by this adaptive and innate responders, if you know what I mean. But we haven't explored much of the ILCs yet, but there is some really good groups in Singapore exploring that. So there is information available for sure.
Yeah, very interesting. So Professor Anderson is congratulating you in the chat.
Thank you, Anderson.
If anyone have a question, you can raise your hands. Gustavo.
Hi, Raíssa. Thank you so much. Wonderful work. I think Anderson has left.
My pleasure.
But he said hi, you know, so on. I have a bunch of questions, but let me ask first something that I probably missed before, is when you compare your CD4 Hanks3 overexpressing with the CD8 regular. So I understood that these were tissue memory CD8 or not, or were circulating CD8 cells?
No, they were all isolated from our transcriptome data. They were all isolated from the skin. So
it should be tissue resident.
Yes, that's right.
Right.
We didn't. We did one thing that we didn't use to separate the epidermis and the dermis. So this is full skin.
Full skin,
so,
right.
Yeah, so the C4 control cells, they are found in the skin, but they are not completely resident, right? Because it was someone published before that they are actually recirculating.
Right. So this normal CD4 that you isolate, it, as I understood, the cells are ranks 1 positive or not, and then the CD8s are ranks 3 positive as well. It's just not overexpressing, but are they ranks 3 positive? Because I understood that ranks 3 is important to drive the differentiation to CD8s.
Yeah, that CD8 population that is resident in the skin, they do express ranks 3.
Right. Okay.
If you look at the CD4 ones that were isolated from the skin, they
don't have it.
Well, they express more ranks 3 than naive CD4 T cells, but they are not as high for ranks 3 as the CD8 T cells. So they're in an intermediate level of expression between naive CD4 and CD8 tissue resident memory. So we believe that the level of expression, so then let's say the number of ranks 3 transcription factor molecules necessary for driving the signature is important here, driving this phenomenon.
Right. Yeah. So that was the point that I wanted to understand well. So all these genes that are, shared between these two populations that you compare, are they ranks 3 targets, like downstream, or not? It's only
not all of them. So
right.
It seems like some of the TGF-beta regulated genes are downstream of rank 3, so they're being directly open. So rank 3 is opening the chromatin structure in those regions, but not all of the genes that are changing are rank 3 dependent. It can be other transcription factors that would be downregulated by rank 3 that now can't transcribe, and now they're changing the expression pattern as well.
That's great. So now one thing that I got this feeling is that these tissue resident cells that you just very well described, and which are with a different shape, like this dendritic cell types, and move slowly, they very much look like activated T cells. All activated T cells in vitro or in vivo, in many different setups, they behave like that. So do you believe that either this set of genes, which I couldn't follow, when you presented, are they associated with activation programs, including metabolic programs?
They would be not all of so the metabolic program of an effector T cell is completely different from the memory one. So they have separate requirements, right? One is more glycolytic, the other one is going to be more dependent on the lipid metabolism. But we do see similar activation markers, right? So CD69 is in reality one of our most, used activation markers for CD8 T cells. And what it does is to hold the cell within the lymph node to promote this activation process better, right? But that's also helping the cells to anchor in this non-lymphoid tissue. So that there are similarities between tissue resident memory T cells and activated cells as well. But they do exist in different phases, right? So while the effector cell would be vanishing at the memory stage in the absence of virus or pathogen, the memory T cells would stay. So I think that the major difference there is the type of metabolism that these cells adapt to, and the differentiation state where these cells are encountered as well. And that is a very, I can talk about differentiation in a very long time. And Dave has this work that Dave has been doing is just so meaningful for this, because if you get, an immunology book, what you see is that how does the immune response work for T cells? So you have naive T cells that are in the lymph node. These T cells get activated, they become effectors, they migrate to the tissue, they respond, and they generate memory. And that's all. Our memory populations that are, sustained long time are believed to be coming from the circulating memory T cell pool. And we call that stemness, right? So it's the stemness capacity of the circulating memory population to maintain memory T cells long term. But what his finds indicate are that these resident memory T cells that were primarily derived in situ, so in the barrier tissue, they can generate, they also have stemness. So they have the stemness characteristic that allows them to generate cells that will fall back in the circulation and are going to be kept long term. So they are actually helping the maintenance long term. And they're, prevalent. If you look in numbers, you have a lot more T cells residing in your peripheral organs than you have in your circulation. So it seems like they're actually major source of this long-term pool of memory T cells that are pathogen specific.
That's beautiful. Just two quick questions now. One is, how far can this tissue memory goes? So you say that it's restricted tissue, like skin. How far can they go? Can they go like millimeters, centimeters, or they travel all way, but remain the tissue? But so how far can they go?
Yeah, that's a very, very good question. I think that it would be within, that's a, a very shameful guess, but centimeters wide. So if you look at a mouse that is immune to HSV in one flank, so in one side, they don't have many tissue resident memory cells in the other side, so in the opposite flank.
Right.
So it seems like they don't go too far away. But when the cells get reactivated, so if you get a reinfection, then you get these cells falling back in the circulation and in the lymph nodes, right? So then you're gonna see a broader dispersion of these memory T cells. And that's super relevant for humans, right? Because a human doesn't get one infection to something, you get like 10 or hundreds over your lifespan. So then what are these cells doing in that situation? How are they redistributing, and what are the phenotypic characteristics and transcriptional characteristics? That's where we are getting into now.
Beautiful. Thanks a lot, Raíssa.
Thank you. Thanks for the question.
So I just want to ask one thing. Do you think that your animal with RUNX3 in CD4 cells have any problem?
We didn't notice in this model that we are using, but one hypothesis that we have is that maybe the CD4 T cells are kept less resident because of the involvement in autoimmunity. So let's say you drive, lots of these protective T cells in the skin, and somehow they prime, other cells that are responding to self-antigens. Does that actually increase
maybe allergy? And I don't know.
Allergy or pathogenism.
Because it must have, I don't know, I think that there's a reason why they are less resident than CD8 cells,
Yes, that's what we think too. And they're also a lot more complex, even though we know that today we know that we can find CD8 cells that are TC17 and TC1s and potentially regulatory ones, but how does that work for CD4 T cells where we know this super broad polarization populations that they can come as? How does that change memory generation? We don't know that, but for sure there must be evolution reason for this to happen.
Okay, very good, beautiful.
Thank you.
So Thiago Francisco asked a question in the chat. He asked, Raíssa, a great presentation. You commented on the phenotypic and morphological differences between resident and circulating memory cells. Could you comment a little more about it? And these resident cells have a long life? If so, through which mechanisms?
So, it looks like their morphological differences are mostly related to their position in the tissue, right? So not all the tissue resident memory T cells are going to look the same. CD8 T cells that are in the epithelial layer, in the gut or in the skin, we see them being this more dendritic-like shape, while the ones that are in the circulation or in the less tight tissues, such as the dermis, they are more brown, so they're basically circular. These tissue resident memory T cells, they do live long term, and you can check. There's a Nature paper from Dave Mazepou's group showing how long these populations last, but basically in mice through the lifespan of the mouse. So as long as the mice live, you're going to have that population. And I think that Rafi Ahmed also has a very interesting study using heavy water in humans, and he can also look at how long these populations are kept. They basically live in tissues by homeostatic proliferation, so there's a self-maintenance of the population in the tissue. And in the case of boost response. So if you have a reinfection, then you're also going to have expansion of that population, which makes their lifespan even longer.
So we have another question in the chat from Liz Dani. She's asking, did you evaluate the production of other cytokines like T helper 2 signature, IL-4 and IL-5 in those cells, or inflows from CD8 are more committed to a T helper 1 profile?
Yeah, they are a Th1 cell type in this case. I didn't try to stain for IL-4 or IL-5. We could potentially go back and look at the RNA-seq data, but the CD4 T cells responding to HSV, so viral infection is driving this CH1-like signature. There's another person with raised hand, Miriam.
Yes, Miriam.
Hello, Raíssa. I'm very sorry. I'm gonna start apologizing because I had to be in transit to get the kids at school. So I'm afraid I'm gonna ask a question you have already answered in your talk. My connection was a little bit unstable, but I had basically two questions. One of them, I remember a long time ago, a big discussion about which APCs would activate the lymphocytes and how they would address the lymphocytes to go back to the site where they came from. And a classical experiment using the dendritic cells from the spleen versus the mucosa from the gut. So one question is, do you know if, depending on where in the skin, which draining lymph node, if they can direct the lymphocyte to a particular part of the skin, or if the skin is a whole organ and they go everywhere?
Yeah, there is a very interesting paper published, I think 2 years ago, showing that the cells that get activated in the skin draining lymph node, they are more prone to TGF-beta imprinting because the DCs are different, right? The DCs are more 103 positive, and they are going to be the ones present in the draining lymph node. So basically, I think there's a gradient of chemokines that will make the cell migrate back to the skin. And we see that there is a preference in migration driven by the draining lymph node of each site. So if you take cells out of the mesenteric lymph node, so that's the one that drains the gut, you're going to see that more cells are going to go to the gut instead of to the skin. But in respect of which draining lymph nodes drive more cells to each area of the skin, I am unsure. And I think that that's based on other, inflammatory gradients that will make the cell migrate back to the site of infection.
Okay, so probably something from the popliteal wouldn't target them to the legs versus the inguinal or some of the neck lymph nodes that could target them to the upper body.
I don't think so. I think it's just the closest one, but I'm not sure how does that impact the distribution.
Okay, and the other question is if do you know if people with psoriasis or with diseases that are based on the skin, if you do find prior to the disease onset, like if there will be something different between the signature of the resident memory cells in one person versus the other, but basically prior to the active disease, do you think that could prone the person towards developing the disease or could contribute to?
I think that this is a very new area. I don't have any data in mind to back what I'm saying, but in my head the way that it could work is, let's say you have a big population of CD8 T cell in your skin driving the response to some pathogen, and once that happens, a lot of cytokines are going to be produced, right? And the cytokines are local, and they're affecting other cells that are unrelated to the infection that's happening. So let's say you have lots of gamma production, and that will change the MHC expression in the tissue, and that could present more self-antigens, triggering an autoimmune response. So it could, lead to, I think, but it's too new, and there is not much published in that area yet.
Okay, thank you. Congratulations on your work. It's really, really amazing. Thank you.
Thank you.
So, Raíssa, I think we don't have more questions for now, also on YouTube. So thank you so much for being here with us today.
Thanks
It was
for
a
the
pleasure
invitation.
for us.
Maravilha, Raíssa. Super, super obrigado. Você está mandando bem demais.
Muito bom. Parabéns.
Vai abrir concurso por
obrigada.
aqui. Então, se você estiver pensando em voltar, fica com a antena ligada. Se você estiver querendo ficar por aí, faz muito bem, que a coisa está feia.
Mas bom saber. Aqui tá bom. Acho que eu vou ficar por aqui por enquanto.
Isso aí, tá ótimo.
Obrigada pelo convite, obrigada por
Obrigado
assistir.
você acordar cedão.
A gente agradece.
Tá preparado aí para contar para a gente essa história, tá bom? Beijão, esperamos te ver aí em breve.
Beijo aí.
Obrigada, Raíssa.
Pessoal, ótima semana para todos. Obrigado a todos por terem mudado o horário também, estarem aqui com a gente. Aquele abraço.
Tchau.
Boa semana, pessoal.